So my conclusions. As I said, normalization has a big impact on differential gene expression. There's many different methods to remove these unwanted variants. It's quite often also an important step to clean up your data and get good separation, because you don't want to cluster based on quality of cells or cell size or percent mitochondria. If you see that, really think about, is this biological? Do I need to remove this? And always I do some sort of selection of variable genes. It might require quite a lot of genes if it's a super noisy data set. And this I haven't added in the slides, but it's also important to think about that when you select variable genes, if you have multiple batches, a lot of the variable genes you select might be batch effect genes if you have a strong batch effect. So you should do variable gene selection per batches. There are also methods in Scran and Scampi, but not in Seurat actually, to block for batch information when you do variable gene selection. And to make it as easy as possible, aim for the same sequencing depth in all samples, because that makes normalization so much easier. And when you run 10x, that also means loading the same amount of cells per sample, because quite often you run one lane or one per sample. And if I've had studies where they had one sample with 10,000 cells, another with 1,000 cells, because that's what they could get out of their patient samples, more or less, and then another with 5,000. And they ended up with very different sequencing depths, very different gene detection per sample, which just increases the batch effect quite a lot. And then normalization will be easier if it's fairly even across your samples. And finally, I just want you, there's so much you can do, but start simple. Start with just normal count normalization, do some dimensionality reduction and clustering, look at what you have. It might not always be the case that you do need to tweak every little parameters and scale everything. It's when you see that you run into issue, you really need to go back and think, how can I re-normalize this to find the signal I want, basically. Okay, any questions?
Can I ask about normalization?